Hi, it's my pleasure to welcome you to today's symposium on Parkinson's disease. I'm Roger Albin. I'm the director of the University of Michigan Udall Center. For those of you who are not familiar with the Udall Center's program, the Udall Centers are special research programs sponsored by the National Institute of Health. Each Udall Center has a specific focus on an important aspect of Parkinson's disease research. The goal of the Udall Center's program is to explore these particular interesting and important areas of Parkinson's disease with the ultimate goal of trying to achieve better treatment of Parkinson's disease patients. Here at Michigan, we're focused on cholinergic mechanisms of attentional-motor integration and gait dysfunction in Parkinson's disease. Now, what this means is that we're specifically focused on a constellation of closely interrelated problems related to disordered thinking and disordered gait and balance, problems that Parkinson's patients very commonly experience in more advanced stages of the illness. And we've formulated an interdisciplinary program drawing from a lot of the expertise across our campus. Now, many of you are probably familiar with the idea that Parkinson's disease involves a loss of dopamine in the brain. And this is an important signaling molecule which is made by nerve cells in the substantia nigra pars compacta. And in Parkinson's patients, these cells die, and they can no longer communicate dopamine to other parts of the brain. And thanks to modern imaging methods, some of which were developed here at the University of Michigan, we can actually identify these kind of dopamine nerve cell changes in living patients and actually make fairly precise measurements of the magnitude of the changes. And this can be shown here in these images where you can see dopamine nerve terminals identified by these very red areas. And you can see that you can easily discriminate a normal young individual and a healthy elderly person, and also Parkinson's disease patients on the basis of how much dopamine nerve terminals are present. Now, in our Udall Center program, we're interested actually in a different brain chemical messenger that's abnormal in Parkinson's disease, and that's a compound called acetylcholine. And somewhat like dopamine, acetylcholine is produced in a small number of nerve cells and then sort of broadcast to many other brain regions. And you can see here in the cartoons on the right, in these sort of red arrows, the bluish arrows, you can see where the acetylcholine is made in sort of small parts of the brain down at the bottom of the brain, and then how it's directed at many other brain regions. We know that acetylcholine is very important in certain aspects of thinking, and this relates directly to our interest in problems with walking and balance. Now, many of us tend to think of walking as a simple sort of automatic activity. However, walking is actually quite a cognitively demanding activity. If you think about it, when you're walking down the street, you have to pay attention to obstacles, but you also have to ignore things that aren't relevant or that won't get in your way. And we've all had that experience of being momentarily distracted and then having a trip or even an actual fall. So there's this close relationship between important aspects of thinking, particularly the ability to pay attention to your environment appropriately, and motor tasks such as walking and maintaining an upright posture. And we know from prior work done over a course of many years that systems involving acetylcholine in the brain are very important for the regulation of attentional function. Now, in addition to its well-documented and specific role in attention, acetylcholine is also involved in a number of other cognitive functions and probably contributes to many of the different aspects of cognitive decline that many Parkinson's patients experience with disease progression. And just as we're interested in how changes in acetylcholine systems influence attention and gait function, we're interested in the general question of how changes in this particular brain system interact with changes in the dopamine system and how those combined changes and interactions contribute to cognitive impairments in Parkinson's disease. This cartoon gives you just an overview of the way we're trying to think about the brain systems we're dealing with. So we think there are many, many interacting circuits that are involved in different aspects of cognition, gait, and balance. And we're specifically looking at to see how these different systems and their interactions are influenced by the different acetylcholine systems. Now, this is a simplified cartoon, but identifies some of the key regions that we're focusing on in the Udall Center's program. And just to give you a brief overview of the way our center is structured, We have 4 projects focusing on different aspects of acetylcholine changes in Parkinson's disease, and they're supported by several cores which provide services that the investigators use for their experiments. Now, a notable feature of our center is that it's quite interdisciplinary in that we have investigators from a number of different backgrounds. So, for example, in Project 1, Dr. Bonin, who's trying to study the evolution of changes in gait and balance in Parkinson's disease, is appointed in the Department of Radiology and also in the Department of Neurology. In Project 2, Dr. Martin Sarter, who is a neuroscientist in our Department of Psychology, is trying to use animal models to understand better how acetylcholine and dopamine systems interact in terms of producing problems like falls. In Project 3, I'm trying to understand cognitive changes over time in Parkinson's patients. And my background is in neurology, and specifically in movement disorders neurology. And finally, in our 4th project, Dr. Omar Ahmed, another neuroscientist in the Department of Psychology, is intensively studying one particular brain region that's involved in the navigation in space. And in our cores, we have support from individuals' expertise in imaging, biostatistics, and other disciplines. And just as we have investigators from a number of different disciplines, we're using a variety of different techniques to address these questions. Now, this is an example of a method that we can use in living humans. This is an imaging method called PET scanning, where we can actually measure and look at the distribution of acetylcholine-producing nerve cells in the brains of living people. And this particular set of images shows the pattern that you would see in normal individuals. We can use this type of information to look at changes in the brains of patients with Parkinson's disease. Now, we can also use animal models to try and do experiments that are really not possible in humans. And for example, one of our investigators, Dr. Sarter, developed an animal model of falls and freezing behavior in rats. This allows us to actually look at the interactions of brain systems and see what's going on. And that was a normal rat crossing this rotating beam and going through a doorway. Now, this is a rat with changes in its brain to mimic what happens in Parkinson's disease brain. You can see it gets stuck at the doorway, and then it actually will fall off the rotating beam. And this is another animal, in this case, trying to cross the beam, and actually gets stuck, frozen at a doorway, just as we see with some of our Parkinson's disease patients. Now, these kind of models give us an opportunity to actually look directly in the brain to see if there are specific changes that we could identify and pursue for possible therapeutic interventions. Now, in the talks that you'll see a number of our younger investigators are going to present some of the work that's been done more recently in the Udall Center, involving both human experiments and also some animal experiments as well. So thank you very much for your attention.